Welcome to this Kabis Impact Formidling meeting. My name is Marit Eggen and I am Project Coordinator and DP Leader of Mobility in Kabis. The intention of the Kabis Impact Formidling meeting is to give industry actors first-hand information on practical implications from recent research. And today we are very pleased to welcome doctoral candidate Enrique Pino Martinez, who will present his work on early maturation. So please, Enrique, the screen is yours.
So then I'll start. So thanks, Marit, for the introduction, and thanks to everyone attending this seminar today. Hope it leads to some interesting discussion afterwards. So I'm Enrique Pino Martinez. I've been a PhD candidate for the last 3.5 years at NORCE Research and University of Bergen. And my main supervisor has been Sigurd Handeland. So today, what I'm going to present, I have just submitted the thesis for defense. So what I'm going to present today is pretty much all the results we have found so far, and maybe some extra results that we have found in some follow-up research that we have just finished. So my thesis has focused on studying the influence of the main production parameters in salmon aquaculture, such as water temperature, photoperiod manipulation, and feed regime on early sexual maturation of salmon, rare up to post-smolt stage in all the time in freshwater. So let's commence with a bit of background. As most of you, I guess, are aware, there is an increasing incidence of early male sexual maturation. In Norway, there is recently a trend towards intensification and towards rearing large smolts, post-smolts in RAS facilities, extending the land-based phase and shortening the seawater phase with the aim of improving some sustainability issues like sea lice or salmon escapees, discharge of organic matter, among other things. But of course, there are the intensive conditions that we are introducing in these facilities are actually causing this increase in early male sexual maturation. So why should we concern about early maturation in the industry? Well, because it represents serious economic risks for salmon producers and also from a fish welfare perspective. Yeah? So when salmon commence to mature, they reduce the feed intake and they use the resources for gonad development. So of course, if we keep putting feed in the system, but the salmon is not eating, then we're going to experience poor feed conversion rate and of course reduce growth. And then there is also a chance for potential osmoregulatory issues that can lead to poor performance, poor welfare, and risk of mortality when the fish is transferred to seawater. And this is because the smoltification and maturation are somehow processes that can be in some physiological conflict because they entail opposite osmoregulatory changes for opposite migrations. So what are the main causes that have been researched so far and related with sexual maturation? Mostly the main production parameters that we manipulate in aquaculture facilities, and this includes photoperiod manipulation. We normally raise, after first feeding, we raise the fish in constant light just to maximize growth, and then afterwards we introduce a winter signal photoperiod in order to induce multiplication. But this winter signal can actually act as a synchronizing cue that triggers onset of maturation instead. Then the high temperatures used in RAS facilities, high sustained temperatures that accelerate and promote fish development, so they can actually attain resources earlier in time and be ready for maturation earlier. Then the intensive feed regimes that we use, the high energy diet supply that cause higher growth rate or attaining larger size earlier in time, again impacting on the fish having enough resources earlier in time for sexual maturation. And then there is other factors. Genetic background is a very important one, and others that actually apply very specifically to RAS because of the high recirculation rate. For example, potential accumulation of sex steroids in the RAS water or other endocrine disruptors. But these last aspects a bit aside of the focus that we have given this study, right? We have focused on the main production parameters. So how did we tackle early maturation in this project? The whole project took place in freshwater. The first experiment, we ran 3 experiments with the whole project. The first one took place in a commercial setting in the RAS facility for post-smolt production that Bremnes Seashore owned in TROVÅG in Rogaland. And there we only looked at the effect of photoperiod on early maturation. Results from this experiment actually pointed more towards other factors like temperature and growth as more related with sexual maturation. So then the experiment 2 focused more on these 2 parameters, on temperature and feed regime that we use to manipulate growth. And we again use fish from Bremnes Seashore, but we transfer it to the facilities, the flow-through facilities we have in Bergen, because we wanted to have more control on the experiment. And then after this experiment, the role of photoperiod was still not clear to us. So we wanted to look at it in correlation with temperature, because temperature we found it was very important. So we decided a 3rd experiment that looked at the effect of photoperiod and temperature. Again, we used fish stock from Bremnes. It was actually the same genetic stock, and we transferred it to Bergen, to the flow-through facilities, in order to have a high control of the conditions. Then the methodology we used in the 3 experiments, we generated time series datasets. This means that we followed the fish for a period of time, and we performed sequential samplings over time. In each of the samplings, we measure body weight of the fish sample and the condition factor. We also opened the fish and extracted the gonad, measured the gonad in order to calculate gonadosomatic index. This index is quite a basic index that only tells us what percentage of the whole body weight is caused by gonad weight. And we use it because when the fish commence to mature, this index starts increasing very rapidly as gonads develop. Then, based on gonadosomatic index, we calculated the proportion of maturing fish at each sampling and in each group. And it was done using GSI as GSI 0.06% as a threshold value. This value was chosen upon previous literature and also upon a statistical study of distribution of the GSI variable that we performed. And this is something that we can talk about later in the discussion if there is interest. Then during the experiment, we also monitor various physiological markers of maturation and of smoltification. Markers of maturation we looked at. We looked at the gonadotropin expression of the pituitary. Gonadotropins are hormones that regulate the onset and continuation of sexual maturation, the main one being FSH. And then we also looked at the plasma 11 keto. 11 keto is the main sex steroid in fish, and is produced in the gonads upon FSH stimulation, and it increases very rapidly as well when the fish commences to mature. And then as marker of smoltification, we use a standard that is a GIL ATPase activity, and we use this one because it increases as well when the fish commences smoltification and commences to develop seawater tolerance. So now let's commence talking about the experiments. We'll go through results and discussing results of each one as we go in. So experiment 1, as I said, it looked only at the effect of photoperiod, and it took place in a commercial RAS facility owned by Bremnes Seashore. So there, this is a diagram that represents what the experiment consisted on. We followed the commercial group that they were producing there. It was the commercial group 721. And we started following them when they were 26 grams in June 2018. And we finished when they were 152 in November, just before seawater transfer. We sampled 5 times, once a month, and then we only had 2 experimental conditions. One group that was kept in constant light, and the other one that received a winter signal to induce multiplication in August and September. What was the main result from this experiment? So it was that we didn't obtain any maturation in the whole group. Neither we nor the staff at TROVÅG that those who are monitoring the group, they didn't find any maturation even. The mean GSI, as you can see here, was 0.041, which is below 0.06 is the threshold that we established, so no maturation. However, we found some signs of reproductive activation, and that's what we're going to look at now. So the first graph above represents gonadosomatic index over time for the two experimental groups, right? So if you look at what GSI looked over time, it was all below 0.06. So it was a fish that had not commenced to develop the gonad, therefore there was no maturation. And of course, there was no effect on the weighted signal, the shaded area. There was no effect on gonad development. As you can see, the two groups had pretty similar gonadosomatic index, so no effect of photoperiod on GSI. However, like in the early samplings, we found a group of fish with some elevated gonadosomatic index that we suspected that they might be engaged in some, like an very early stages of sexual development. So we wanted to know a bit more about those. And then in order to find out more about that, we analyzed FSH in the pituitary, the main gonadotropin kickstarting the maturation process. And we found that indeed in June, we had elevated levels of FSH, but then these levels significantly decreased then in July, and then it continued to be low for the rest of the experiment, and no difference between the two photoperiod groups. So again, revealing that the change in photoperiod did not have any effect on sexual maturation. And we only had some slight signs of sexual activation, sexual development in June, when the fish was still in long light, in continuous light. So what happened here, we believe it's called as a dummy run. And a dummy run is just a slight sexual activation, an activation of the brain pituitary gonad axis that can happen, but that does not lead to complete maturation. And it can happen only after exposure to certain environmental conditions. And how do we explain the occurrence of this? Well, this graph shows the mean weight over time of the whole group and the temperature profile of the whole group. This was data from TROVÅG. What we found here is that this group, the whole group, experienced quite a steady increase in temperature from February till June, from pretty much 4 degrees to 16 degrees. So most likely this increase in temperature caused some sort of reproductive activation that caused a bit of upregulation of FSH production and a bit of gonad development, but it didn't lead to maturation. So why didn't it lead to maturation? And to find that out, we asked TROVÅG if they could supply us with some more data from the previous groups that they had raised in the facility before. So they gave us data from 5 groups that they had produced there before. Groups ranging from 717 to 721, that is the group that we followed. The data that they gave us were mean body weight per month for each of the groups, mean temperature per month for each of the groups, and then the final percentage of maturation that they had estimated in each of the groups at the end of the cycle before transfer to seawater. So as you see, there were 2 groups that they found they had a 10% maturation, 2 groups that they found no maturation, and 1 group with 5%. There's this group is the one we followed, it had no maturation. So with this data, what we did was generating a different data set, calculating several variables that we did ourselves. And these variables were calculated for each of the groups for the whole production cycle. So these variables were the weight gain, the mean specific growth ratio that each of the group experienced in the facility, the mean specific growth rate for the first 5 months, the temperature zone, the mean temperature only for the first 5 months, and then the temperature range, or the difference between the maximum and the minimum temperature that each of the groups experienced. With this data and the percentage of maturation, what we did was running a Pearson correlation coefficient matrix in order to find out correlation between variables and percentage of maturation. And what we found is that mean temperature was the variable that was more related to percentage maturation. It was a very high correlation. So 90% of the times that we found high maturation, it was linked to high mean temperature as well. And then closely followed by mean specific growth rate. So mean specific growth rate, also very important for maturation. Then in order to find out more about this data set, we run a principal component analysis. This is just a statistical tool that tells us about the structure of the data set, about the correlation between the variables and the correlations as well between the variables and the groups. The only thing we need to know here is that the direction that the arrows are pointing towards indicate the direction in which the variable grows. So if we look at the location of the groups in the graph, let's look at these two groups, 717, 718. Both of them had 10% maturation. That's why they are in this area where maturation is pointing towards. This area is also characterized by high average temperature and high specific growth rate. So high maturation, as indicated here, linked to mean temperature and mean specific growth rate. Then, if we look at the group that had 5% maturation, is located in this area of the graph, middle area, that is characterized by mid values of specific growth rate and mid values of temperature. As a result, we had mid values of maturation of 5%. And finally, we look at the two groups in the facility that did not mature. So we look at the 719 located in this area. This area is opposed to this in the graph. So therefore, this area is characterized mostly by lowest specific growth rate. This group experienced the lowest specific growth rate among all, and comparatively lower average temperature as well. The result is no maturation. And finally, the group we followed, which is 721. This group is located in this area that it's characterized by the lowest average temperature of all and the highest temperature range, so highest oscillation of temperature. So based on all that we have said about experiment 2, we can conclude that first, photoperiod did not seem to have any effect on sexual maturation in this experiment, in the trial that we performed. And in the facility, including all the groups that they had raised there, maturation appeared mostly linked to high mean temperature and high mean specific growth rate. And these actually suggest an explanation for the lack of maturation in our experiment. And this is because this group, the group we have here, experienced the lowest average temperature of all groups, comparatively low specific growth rate. So most likely this group did not develop sufficiently, did not attain enough resources for maturation when the winter signal was introduced, and therefore they couldn't mature. So, okay, then we have, from here, we take home message is that high temperature and high growth seem to be the most important factors for maturation. Then we designed an experiment 2 that looked at indeed the effect of temperature, and we use feeding regime to manipulate growth. So here we have a diagram of what the experiment consisted on. It was a 3 × 2 factorial design. We used 3 temperatures, 18 degrees, 12.5, and 8 degrees. And then 2 different feeding regimes. One group that received a full ration, and the other one that received 67% ration at each temperature. The experiment ran from October 2018 to June 2019, and we sampled 8 times plus an endpoint at the end, where we assess only percentage of maturation. And we introduced a winter signal here in February and March in the shaded area, just to induce some developmental response, whether smoltification or maturation. The main result in this experiment was that we obtained a lot of maturation at 18 degrees, pretty much 100%, and remarkable degree of maturation at 12.5 degrees, while no maturation at all in 8 degrees. So let's have a closer look at that. And this graph is showing the percentage of maturation over time for the 6 groups. Starts from late March. This is the sampling file here, because this is when maturation commenced. So we see that it was in early April when we first found some fish maturing happening in the two 18 degrees groups. And from that time on, the proportion of maturing fish continued to increase until the end of the experiment in late May, where we pretty much had 100% of maturing fish in the two 18 degrees group, regardless of the feeding regime. Then at 12.5 degrees, maturation was much lower. Within the 12.5 degrees group, maturation was much higher in the 12.5 full, where we had a 40% maturing fish at the end. There was much less proportion of maturation in the 12.5, 67% ration here, just a 9% maturing feed, but still present despite the feed restriction. Yeah? And then finally at 8 degrees, as I said, there was no sign of reproductive activation at all. So it seems very clear that that temperature had a primary effect promoting sexual maturation, right? But then what was the effect of restricted feeding on maturation? So to find that out, what we did was establishing 4 maturity categories. As you see here, they range from immature, early stage, maturing, and mature. It was based on different GSI values, somatic index values. With these maturity categories, we calculated the percentage of each of them in each sampling for each group, and then we plotted the cumulative percentage of maturation. And this is the graph we have here, and we're going to go through now. So if we start with the group that had the least intensive conditions that had maturation, so this is 12.5 and restricted feeding, we see that maturation only happened in late May. It was at a small percentage of fish, and it was mostly represented by early stages. Then, as intensification increases, we move on to the next group, it's 12.5 full ration. We see that maturation commences earlier in time. We have higher proportion of maturing fish, and we have all maturity categories represented. And then, if we move on to the last 2 groups at 18 degrees, we see that maturation was so much higher. It happened earlier in time, and we reached in both groups pretty much 100% of fish maturing. However, there was some difference between these 2 groups, between the feed regimes, and is that in the group 1867, the process seemed to happen a bit more gradually. As you can see, all the maturity categories were more evenly represented as time went on, while in the group 12.5 and full, it was mostly more advanced stages of maturity from the very beginning. Okay. So we have seen that high temperatures seem to be the most important factor for maturation. But how did it happen? What were the mechanisms causing this activation of the reproductive axis at high temperature? We think there were 2 mechanisms. The first one, we think it was faster energy acquisition at high temperature. So it is known that at higher temperature, the fish has an accelerated metabolic rate. They show more appetite. They acquire energy more efficiently. And if we look at this graph, it shows the condition factor over time for the 6 groups in this experiment, the full ration, the 67% ration here. We see that condition factor was significantly higher in the 2 groups at 18 degrees from the beginning till the end of the experiment in the 2 feed groups, right? The 2 feeding groups. So this is an even before and after the winter signal. So this is suggesting that these fish had actually gained energy at a faster rate that they could use afterwards for sexual maturation. Then another process promoting early maturation at high temperature was an early activation of the brain pituitary gonad axis or the reproductive axis from brain to gonad. Let's look at a closer look at this. Up here we have the gonadosomatic index over time for the 6 groups. So the first thing we noticed was that the gonadosomatic index was significantly higher. This was shown by a statistical model at 18 degrees in the 2 18 degrees groups, regardless of the feed regime, even before the winter signal. So from very early in the experiment. So these gonads were more developed from very early in the experiment, meaning that they could be ready for maturation, of course, earlier than the other. And this happened before the actual onset of maturation, which happened after the winter signal. Here, we have this sudden increase in gonadosomatic index that is showing that the gonads had commenced to develop very rapidly, and the spermatogonial proliferation has started. So we had an early activation here before the winter signal, and this, in order to find out if that was supported by the endocrine regulation, then we looked at the pituitary FSH expression. And what we found is that indeed, there were also higher FSH transcripts that were present in the 218 degrees groups before the winter signal, supporting actually the larger gonad development found here as well. So this is clearly showing that there was an early activation of the brain pituitary gonad axis at high temperature. And we have plenty of more data of other analyses that support this early activation at high temperature. We have support in gonad analysis that we have performed. For example, during this period, we have found downregulation of a hormone produced in the gonads, anti-mullerian hormone, that is an inhibiting factor. So when it's downregulated, it allows maturation to proceed, to go on. And we found it downregulated already at this stage. Then also at this stage, we have found presence of more advanced germ cell stages, which means that these gonads were more developed than the other ones in the previous groups. And also we have found signs of neuroendocrine activation of the BPG axis, like upregulation of gonadotropin releasing hormone, or in the case peptine. This is, if there is interest, we can talk about more. We can talk about this more afterwards. Then what happened with smoltification? This is the GIL ATPase activity over time for the 6 groups in this experiment. The first thing we noticed was that there was a very low NK activity over time at 18 degrees, regardless of the feeding regime. It didn't have any effect, the feeding regime here, before and after the winter signal. So it seems that the high temperature had definitely a disruptive effect on the physiology, impairing the function of this GIL enzyme. It could be a direct impairment of the function of the enzyme, or could also be a negative interaction between smoltification and maturation, because again, these processes may present some sort of physiological conflict. However, this is very difficult to tell here, because pretty much all the fish at high temperature mature. Then on the other end, on the contrary, we had the groups at 8 degrees that were the only ones that showed clear increase in NK activity after the winter signal, indicating that these fish had commenced to develop seawater tolerance, right? And then finally, the groups at 12 degrees, we found some slight signs of there was high variability in here, with some elevated values of NK activity, meaning that most likely these fish may have developed seawater tolerance just by size, just as part of their continuous development, because at this stage, this fish was already 150 to 200 grams. But after the winter signal, these fish did not increase NK activity in the gill, and actually it was mostly a decreasing pattern, and they showed a tendency to sexually mature. So what can we conclude based on all that we have said regarding experiment 2? Well, we can conclude the first thing is that high temperature clearly was the main factor promoting early maturation, and this seemed to happen through 2 different mechanisms. One that is accelerated development and more energy acquisition, and the second one being an earlier activation of the reproductive axis. Then the feed restriction only modulated the process. At high temperature did not have a major effect. It had a larger impact on when the temperature was lower at 12.5, but still at that temperature, it did not impair maturation. Then, as we see in the graphs above, high temperatures seem to impair development of seawater tolerance. And this may have happened through a direct impairment, just impairment of the physiological function of this enzyme, or because of conflict, physiological conflict with maturation. But again, this is difficult to tell here because it's confounded by the fact that all the fish at high temperature mature. And then from this experiment, we couldn't conclude on the role of photoperiod. And this is because we introduced the winter signal in all 6 groups. And therefore, even though the onset of maturation appeared to commence right after returning to 24 hours from the winter signal, we couldn't conclude such thing because we introduced the winter signal photoperiod in all groups and we didn't have a control group. So because of that, and because of finding the high temperature was so important, we decided to run a 3rd experiment that looked at the effect of photoperiod in combination with temperature. This is the diagram of this experiment. This time we used 15 degrees at 12.5. We brought it down from 18 to 15 because we thought that 18 was quite extreme. And at each temperature, we had 2 photoperiod groups. One that was kept in constant light, LL, and the other one that received a winter signal at the same time as the previous year for the same duration as well. For this experiment, we used the same fish stock as in the previous one, same genetic background, and it ran from November 2019 till May 2020. We sampled 8 times during that period. The main results, one more time, the very clear effect of high temperature. We found higher maturation at 15 degrees. We can see it in this graph. This is a percentage of maturation over time for the 4 groups of this experiment. And what we see is that in the 2 groups at 15 degrees, we had higher percentage of maturation. 100% of the fish was found maturing at the end of the experiment in the group 15 winter signal, while 75% was found maturing in the group 15 LL. And then at lower temperature, similar pattern, but just lower proportion of maturation, simply because of the less stimulatory effect provoked by 12.5 degrees, right? We have 50% of fish maturing and 12.5 winter signal and 25% of fish maturing here in the 12.5 LL. So then, again, very clear effect of temperature, but then what was the effect of photoperiod? So we found that the effect of photoperiod was most likely acting as a synchronizing cue that entrained onset of maturation with fish inner rhythm. So they knew when to commence the process of maturation based on the change of photoperiod. And this effect was very large in highly stimulated fish. This is salmon that had been reared under very highly stimulatory conditions on high temperature. So we can look at that by looking at gonadosomatic index again over time for the 4 groups. What this graph is showing is that the gonadosomatic index was already elevated in 15 LL here in February. And then from here and on, it continued to increase until the end of the experiment. But this increase happened like with very large variability, very large intrasampling variability among individuals. Right? That's why we have this huge standard error. And bear in mind that this is a logarithmic scale. So if it wasn't a logarithmic scale, this standard error would look even larger. So this means that at each sampling point in this group, we had fish that displayed very high gonadosomatic index. This is very highly developed gonads, or very low. And this happened as time went on. So what this tells us is that this was not a synchronized process. This was just a process happening spontaneously in some individuals and not happening in others. And that was the only thing here. We had an opposite effect in the group 15 winter signal. We see that the dashed line in red, there is no elevation of GSI during the dark period. So the dark period actually blocked gonad development. But then when the fish is returned to 24 hours, there is a sudden increase in gonadosomatic index. This is the gonads commence to develop and maturation has started. But this increase actually occurs in a highly synchronous way. As you see, there is very small standard error. So this is very small intrasampling variability among individuals. This means that all the fish in each of the groups were presented the same stage of gonad development. And therefore, they must have started maturation all simultaneously in response to the same photoperiod Q. And this Q was, of course, the change of photoperiod. Then we had a very similar pattern in GSI variation in the two groups at 12.5, with a small standard error in the winter signal group at 12.5, a larger standard error at 12.5 and continuous light. But overall, lower gonadosomatic index simply because of the less stimulation that these fish had experienced due to the lower temperature. So, and then if we looked at some endocrine regulators like FSH, it looked exactly the same. It showed exactly the same. So FSH, this is the graph, as you can see, resembles the GSI graph very much. Again, in 15LL, we have a very large standard error over time, showing a non-synchronized response. And on the other hand, we have a very synchronized response in the 15 winter signal. And this is just an example so I can show what I mean with very developed or non-developed gonads at all. This is a very maturing fish found in the last sampling here, showing very high GSI. Gonads are very developed, very close to spermiation, almost producing spermatozoa. And then in the same sampling, we had fish that were completely immature. Then what happened with smoltification in this experiment? Well, let's have a look at it. We have here NKA activity over time for the 4 groups. And the first thing we noticed is that in late January, before the winter signal, we had some signs of like large variability as well here. So this means that some fish at 12.5 displayed high NK activity while others displayed low. So this means that some individuals had already commenced to develop seawater tolerance spontaneously, like after attaining a certain size or as part of their continuous development. And this makes sense because these groups at this stage were already 200 grams on average. Then afterwards, the 2 groups at 12.5, regardless of the photoperiod, if it was in continuous light, if they received the winter signal, doesn't matter, they had a decreasing pattern in NKA activity, meaning that if there were some fish that had commenced to develop seawater tolerance, they afterwards de-smoltified. So they lost this type of smolt characteristics and instead showed a tendency to sexually mature. And then at higher temperature, at 15 degrees, what we found was, as in the previous experiment, just a very low NKA activity over time throughout the experiment. So again, revealing either a negative effect, an impairing effect of high temperature on the function of this enzyme, or a negative conflict with the physiology of maturation that was pretty much ongoing here from quite early. Okay. So then, based on this, what can we conclude regarding experiment 3? The first thing, one more time, is that high temperature was the main factor promoting early maturation, right? Then the photoperiod switch from winter signal to constant light acted as the synchronizing cue for sexual maturation. Then high temperature, as we saw here, impaired development of seawater tolerance, and the winter signal did not induce multiplication, but actually maturation. And then from this experiment, we concluded that we had to look at the role of size, of the fish size, when this winter signal is introduced, because we believe that most likely the proportion of maturation that we would get would not have been the same if we introduced the winter signal where if the fish was 60 grams or if the fish was 100 grams, and as if the fish was 200 grams, as it was in here, because of course at 200 grams, the fish has spent more time in a highly stimulatory environment for maturation, and they had the chance to acquire more energy as well. So this led to a 4th experiment that we have just finished, and we have interesting preliminary data that we can also talk about afterwards, if you are interested. So then, this was all the work we performed in the PhD. So what can we conclude based on this? Well, the first thing is, and the main thing I think, is that high temperature, it's clearly the main factor that stimulates the occurrence of early maturation. We think this happened through two main processes: the early activation of the reproductive axis and through higher energy acquisition. And as we saw, the effect of high temperature can be very strong, and it can override the lack of energy availability. As we saw in experiment 2, you may restrict the energy availability in the fish, but they still mature because they are at high temperature, or the absence of synchronizing cues, as we saw in the experiment 3 in the groups that were under continuous light, that despite not receiving a synchronizing cue, they also mature because of the high effect, the high strong effect of high temperature. Then a feed restriction only slightly reduced speed of the process at high temperature, as you saw. Small effect, only kind of modulated the speed at which the process happened. Then at moderate temperature, at 12.5, the feed restriction had a stronger impact on the proportion of maturation, but still this was not sufficient to avoid maturation. Then in highly developed fish or fish reared at high temperature, as we have seen, a winter signal photoperiod and then a switch to 24 hours can actually act as a synchronizing cue and trigger onset of maturation. However, that same fish, if they are highly stimulated and they have developed at fast rate, they may mature in absence of such photoperiod cue. And I think in this case, there is a more important role played by the genetic background of the fish. Then we saw that high sustained temperature affected smoltification. It happened in experiment 2 and 3, but it happened differently whether the group was at high temperature or at moderate temperature. So in the two high temperature groups, at 18 degrees, at 15 degrees, there were most likely two mechanisms. There was some sort of direct physiological disruption of the NKA enzyme function in the gill, and a negative conflict with the endocrine regulation of sexual maturation. But again, this was difficult to tell because most of the fish at high temperature mature. Then at lower temperature, at 12.5, we also found post-multiplication indicators, right? But this may have been caused by a combination of factors, we believe. And these factors could be the fact that first, they all seem to develop high postsmolregulatory capacity, seawater tolerance spontaneously, mostly as they grew, because they reach a certain size between 150 and 200 grams. But then this is a large size, and most likely this means that we introduced the winter signal too late, when the fish were too large, and therefore we missed the smolt window of the optimum period for smolt migration, and therefore this smolt window is thought to be about 200 degree days after peak NKA activity. So we clearly missed it, which means that most likely these fish dismoltified. They lost some smolt characteristics like NKA activity. Then, in addition, this fish was kept in sustained moderate temperature, and at this temperature it has been found that there is a quicker loss of osmoregulatory capacity. So this fish lost some capacity for hyposmoregulatory ability that they might have developed early here, but they lost it here because we kept it at this temperature. And finally, we also kept them all in fresh water throughout the experiment. So this means that they missed the inducing effect of exposure to seawater. So, with considering these conclusions, what are the implications of this for industry and for the current strategy of rearing larger smolts? We have to say that producing larger smolts using high stable temperature and using artificial winter signal increases the risk of early maturation, as well as poor seawater tolerance. So based on our findings, it seems prudent that in salmon production, especially in the land-based phase, the temperature is kept below 12.5 degrees. Because as we have seen, the higher temperature sustained over time can act as a very highly stimulatory factor that will cause an activation of the brain pituitary gonad axis and also promote high energy acquisition, and therefore increasing the risk of maturation and actually not having a positive impact on growth, because we found that the fish at 12.5 actually grew better than the ones at 18, and similar to those at 15 degrees. Then a restricted diet is not sufficient to stop maturation, especially at high temperature. It's not sufficient at all. It doesn't have a strong impact, but it significantly reduces growth. So my guess is this is not acceptable from an economic perspective, and also not from a freshwater perspective. Then the introduction of a winter signal introduced to induce multiplication actually can synchronize the onset of maturation instead. And this is especially risky when we are dealing with highly developed salmon that has experienced highly stimulatory conditions or high temperature. But as we have seen, this process can also start spontaneously under continuous light if the fish experienced very stimulatory conditions. Then I think this is also very important. Early maturation may not commence in the land-based phase, but it could occur then when the fish is transferred to seawater. If this happens, for example, in late fall, you transfer to seawater highly developed fish with a lot of energy. They experience the winter-like conditions in the sea pen, and then they may mature the following spring as grills. And then, in regard to development of seawater tolerance, we have seen that it's possible that if we keep the fish at high temperature, they're going to show poor seawater tolerance, and or if the fish is up to large size, large postsmolts, and kept in fresh water. These fish, these large postsmolts, may desmoltify, and their performance or the welfare in seawater is not that certain. Although there is studies showing that these fish is reasonably robust and they can adapt the osmoregulatory physiology quite quickly if you transfer them to seawater, and they may re-smoltify. But we need to do more research on this ourselves. And there's also, as we have seen, if we work with high temperature, there's risk of physiological conflict between smoltification and early maturation. So, okay, I think, yeah, this is all I wanted to tell you today. There's more things that we can talk about, like future research plans that we have based on the research gaps that we have been identifying as we go on with our research. And the findings from the 4th experiment that we have finished based on size, and just whatever is not clear. So we can talk about that later if it's interesting for you. So thanks for your attention, and Marit, you take on.